Hello, and thank you for watching this presentation by Ibab and Othman, Francis Pick, Asmasaka and myself about toxicity of polycyclic aromatic hydrocarbons to phytoplankton. These chemicals have been known to be toxic to life for many years, and 16 of them are included in priority pollutants. Numerous studies dealing with the toxicity of PAH to aquatic organisms have been performed, yielding extensive ecotoxicity data, including effective concentration values. If we focus on EC50s, we can observe that phytoplankton and microalgae only represent 10% of the available data, for which further examination is needed. Exposure to polycyclic aromatic hydrocarbons have many different toxic effects on phytoplankton. At the population level, growth is reduced and interspecies competition is altered in complex communities. DNA can be damaged and gene expression regulated by exposure. Metabolic changes can occur, for example, in oxidative stress response or photosynthetic activity. Thus, many studies have provided evidence of different modes of action on different biological models exposed to different chemicals. This requires a synthetic analysis of PAH toxicity assessment, where species sensitivity distribution can be useful. Reported range of EC50s is very high, from less than 1 microgram per liter to tenth of milligrams per liter. We selected from the databases in the literature a total of 183 EC50s, processed to draw SSD curves. The harmful concentration for 5% of the species was determined at 3.4 micrograms per liter for all PAHs considered. But many studies are weakened by the lack of chemical robustness, especially in actual exposure concentrations. Furthermore, evaluating a single chemical is difficult due to the scarcity of data. Nevertheless, we can provide a toxicity ranking for 7 PAHs out of the 16 list, naphthalene being the less toxic one, and benzo pyrene the most toxic. A weak relationship can be drawn between toxicity and solubility, the more soluble PAHs being less toxic. Freshwater microalgae appear to be more sensitive than marine species, whereas diatoms are more tolerant than chlorophytes. But the determination of exposure concentrations is often poorly supported in most available studies. This recommends for further experiments addressing those response models of phytoplankton exposed to polycyclic aromatic hydrocarbons. Recent methods, such as passive dosing, might be well suited for such a purpose. Thank you again for watching.